Have you ever wondered how cells divide? What magical process allows life to multiply and reproduce? It's a symphony of nature, a marvel of biology, and it's happening right inside your body every second. This intricate dance of division and multiplication is called cell division, and it's the heart of growth, repair, and reproduction in multicellular organisms. So strap in and prepare for an enlightening journey as we unravel this mystery and dive into the fascinating world of mitosis and meiosis. Scene script. Imagine a cell as a city, bustling with activity. Now this city needs to duplicate itself. But how? Well, let's dive into the world of mitosis, the process that enables cell division. It all begins with the prophase. Here, the cell's chromatin condenses into chromosomes, each made of two chromatids held together by a centromere. As the nuclear envelope breaks down, spindle fibers from the centrosomes can interact with these chromosomes. Next comes metaphase. The spindle fibers attach to the centromeres of the condensed chromosomes and line them up at the cell's equator, between the centrosomes. At this stage, the chromosomes are at their most condensed state. Then we move into anaphase. The centromeres split, allowing the sister chromatids to be pulled towards opposite poles of the cell. The kinetochore microtubules shorten, moving the chromosomes polewards. Finally, we arrive at telophase and cytokinesis. The nuclear envelopes reappear, encapsulating groups of chromatids clustered at each pole, forming the daughter nuclei. The cell elongates, and the plasma membrane pinches inward. The cell's cytoplasm and organelles split, resulting in two identical daughter cells. And just like that, from one, we have two identical cells, each carrying the same genetic information. Now what if a cell needs to divide, but this time, the daughter cells need to have only half the genetic material? Enter meiosis. Let's dive into the first part of this process, meiosis 1. It all starts with prophase 1, where homologous chromosomes pair up and undergo a process called crossing over. This is essentially a genetic exchange program where segments of DNA are swapped, creating new combinations of genetic material within each pair of homologous chromosomes, or tetrads. Next up, we have metaphase 1. Here, these tetrads align themselves at the equator of the cell, each attached to spindle fibers from opposite poles. It's like they're getting ready for a tug of war, and the tension is palpable. Then we move to anaphase 1. The homologous chromosomes split, and the maternal and paternal chromatids start migrating to opposite poles. It's as if the tug of war has ended and each team is retreating to their respective corners. Finally, we reach telophase 1. The nuclear envelope reforms around two sets of chromosomes, each with a different combination of maternal and paternal genes. But hold on, there's no replication yet. Meiosis ends here with two haploid cells. Now onto the second act, meiosis 2. During prophase 2, the nuclear envelope dissolves again and spindle fibers emerge. The chromosomes, already duplicated from meiosis 1, prepare for another round of division. In metaphase 2, these chromosomes align at the cell's equator, each with two sister chromatids, still attached at the centromere. The stage is set for another round of separation. Anaphase 2 sees the centromere split and the sister chromatids start their journey towards opposite poles. It's like watching a mirror image of what happened in anaphase 1. Finally, we reach telophase 2 and cytokinesis. The nuclear envelope forms again, and the cell splits, giving us 4 haploid cells. Each of these cells carries a unique set of genetic material, thanks to the crossing over and recombination that happened way back in prophase 1. And there you have it. From one cell, we now have 4 unique cells, each carrying a unique set of genetic material. So, we've traveled through the intricate processes of mitosis and meiosis. Mitosis, the process that allows somatic cells to divide for growth, repair, and asexual reproduction, takes us through 4 fascinating stages: prophase, metaphase, anaphase, and telophase. In contrast, meiosis, which occurs only in germ cells, involves 2 successive divisions and leads to the formation of haploid gametes. These 2 processes, while fundamentally different in their function and outcomes, share common elements. Both involve the condensing of chromatin into chromosomes, the alignment of chromosomes at the cell's equator, and the splitting of centromeres. They also share the magic of reformation, the nuclear envelope reappearing, and the splitting of the cell cytoplasm and organelles into new cells. Our journey through the world of cell division ends here. But remember, these processes are happening within you right now, enabling life as we know it.